Welcome to clickmyproject.com. Efficient feature selection and classification of protein sequence data in bioinformatics. Now see about the abstract of our project. The proposed technique to select the most significant feature which led to improve the classification results on various kind of superfamily sequence obtained from the publicly known benchmark data sets. Our project we have three kind of steps involved in it. First of all, we have to select the data set from the protein data set, and then training and testing the system, and the encoding of protein sequence, and then sequencing encoding by the combinations of NRAM descriptive frequencies. Because we have 20 standard amino acid in the protein sequence, and find the amino acids could be constructed and investigated the feature of lengths and NRAM descriptors less than, bigger than, and 3. And here we have proposed technique is very simple, fast and reliable and robust, and require a very short training times. This methodology begins with the selection of training data and then proceed with the sequence encoding and feature selection models. In our project there are 4 steps are involved. The first steps we have to select the variable length protein sequence, and second sequence encoding with the combinations of NRAM descriptive frequency based encoding method, and then feature selection with the statistical metric based technique and training a classifier and train classifier. The second is a testing phase. In testing phase, selection of variable length protein sequence and sequence encoding with the combination of NRAM descriptors and feature selection, and then finally classify the trained classifier, and finally evaluation the classification results. Our main concept is to extract the protein sequence combinations and extracted the sample sequence for another protein sequence. We have to find out the different type of protein sequence from the protein data sets. And next we see about the flow diagram of our process. First we have to load the protein data set, and second encoding of protein sequence. We have encoding the protein sequence with the combinations of 20 standard amino acid. The amino acids contain the 20 variables, and we have to provide us a gene, and we have to split into 3 level. For first level descriptor, and second level descriptor, and finally a third level descriptors. And we have to combinations of encoding sequence. What are the combinations are involved in our amino acids? In first level descriptor they have only counting, and second level descriptor we have to combine with the 2 genes in our descriptors, and finally the third level we have to combinations of 3 level descriptors, and we have to assign the feature selection process. In our feature selection process we have to calculate the mean variance, and the vector distance are also calculated in our feature selection process. And finally we have performed the process classification and extract the performance results. This is a flow diagram of our project. Now we have to run our project. First we have to run the home page. Click run file. We have to browse the protein sequence data set is a uniprot FASTA format, and count the number of records in our data set, and then also show the size of the data set. Our project size is 27137792 bytes, and we have selection processes. The selection is completed, and I click the view buttons. The view selection results. Our process takes some time to view the results because it have bytes of data. This is our values, and this is a protein sequence from our data set, and click next buttons to performing a encoding process. The sequence encoded successfully and view the first frequency encoding. This is first frequency encoding to count the protein sequence and divided by overall count sequence. To find the first frequency, this is a value for values for each protein sequence. And I click next buttons, and we have to count the second sequence. And show sequence. There are 401 sequence and is the N2. This is our we have combining the two sequence ordered and have to recommend that this data set is a protein sequence for 2-level data. And click next button and performing the sequence encoding process for second level successfully, and click OK button and then view the second frequency encoding. The second frequency encoding is calculated is easy method because if our sequence encoded sequence protein sequence encoded is x equal to a c d e f g h I, etc. In the second order the first order n level you only calculate a occurs whatever a time in our data set and counting that and divided by overall count. In the second level data set we have identified the combinations of 2-level orders and we have counting the 2-level count. If like AC, we have counting that AC order is how much time occurs and divided by overall time. We have to identify this second order encoding for overall process and click next button. Now we have calculated the mean value for first sequence and second sequence. We have to calculate the Ni. Ni is a overall sequence for first and second. In our first sequence there are 20 sequence and in second we have 401 sequence. And then we give the summation for vector for summation 1. The summation 1 for first sequence is 38151.227. The summation is calculated for overall summations of each protein sequence and click second sequence. It takes some time to produce the result. And the summation 2 result is occurs and click the mean vector. These are overall simulations for identifying the mean vectors. First we have identified the total sequence and summation vectors, and then finally we have find out the mean vectors. The summation vector is identified by counting the overall sequence in proteins and click the next button. Now we have calculated the variance for first sequence and second sequence and select the Ni minus 1 is a total sequence minus 1 and click the summation. Summation result is a means and inverse calculation for summations and click the mean vectors, the variation. So the overall simulations are identified in this frame and click the next button. The vector distance to find out the vector distance from the result of means and variance. So we get the total vector distance is 421 is a counting of first order sequence and the second order sequence, and finally we have generated the vector. Generate is calculated successfully and show these vectors. So we have get the results of vector and matrix for the protein sequence. So we have easily identified the protein sequence where to calculate the vector and can be show in this vector format. This is our future reduction mechanism where it get minus result we have reduced and then we have positive result is the protein sequences get originally results. This is a classification mechanism for to identify the sequence mining for classification approach. Thank you.